Hi friends, so in this video we are going to talk about beta oxidation of fatty acids. So in my previous video I have talked about the synthesis of fatty acids from acetyl coenzyme A, and in this video we are going to talk about the reverse of synthesis of fatty acid, which is the breakdown of fatty acids into acetyl coenzyme A. And this pathway is called the beta oxidation because beta carbon is always oxidized and released. So please make sure to like, share and subscribe my channel. So what is beta oxidation? It's the breakdown of fatty acids to acetyl coenzyme A. So suppose we have a 16 carbon molecule or 16 carbon molecule of like palmitic acid, which broken down to the acetyl coenzyme A. So acetyl coenzyme A is a 2 carbon molecule. So by breaking down one fatty acid, the palmitic acid, we get 8 molecules of acetyl coenzyme A. And this acetyl coenzyme A can directly enter into the Krebs cycle, which reduces lot of energy. And or they can also enter the ketogenesis pathway, where the ketone bodies are produced during the starvation condition and like to supply energy to the brain and other tissues which cannot produce ketone bodies. And where does it occur? So it occurs in the mitochondria because most of the enzymes that we require are present in the mitochondria, and it occurs in aerobic condition because you require like NADH, FADH to get to this reaction. So it mostly occur during the aerobic cycle. So beta oxidation, it involves three stages. First, because it needs to be broken down, like we have a only fatty acid. So the fatty acid needs to be activated. I will talk about in detail in my next slides the activation of fatty acids which occur in the cytosol. And then from the cytosol, the activated fatty acid get transported into the mitochondria by a shuttle system which is called the carnitine shuttle system. And then once it get transported to the mitochondria, then everything else happens in the mitochondrial matrix because the enzymes are present in the mitochondrial matrix. So first we will talk about the first step, which is the activation of fatty acids. So we have this fatty acid which needs to be converted into the acyl coenzyme A, which means we need to add the coenzyme A group to the fatty acids to get transported and then be converted into acetyl coenzyme A. So first what we need to do is to add the AMP, which occur by an enzyme thiokinase. So if you see, this reaction utilizes ATP. That's why the condition needs to be aerobic for this reaction to occur. And then from fatty acid, it can convert it to acyl because this is an acyl group which is formed adenylate. So from fatty acid, we get acyl adenylate, and then this AMP get released by the coenzyme group, acyl coenzyme group, and it adds a coenzyme A at the CO group, which forms an acyl coenzyme A. So because the acyl coenzyme A, this step occurs in the cytosol, and now we need to transport this to the mitochondria. But then there is a problem. The acyl coenzyme A cannot be transported because the shuttle that is present, it cannot transport the acyl coenzyme A directly. So what we need to do is to convert this to acyl carnitine by the enzyme carnitine acyltransferase. So this is the rate limiting enzyme. So what happens in this reaction is the coenzyme group get released and this acyl group get added to the carnitine. So this coenzyme A will get released and then this acyl group get added to carnitine, and then this acyl carnitine can be transported to the mitochondrial matrix because this shuttle system can only bind these and transport these, not these. So when we transport these, the acyl carnitine again get released the carnitine and we get acyl coenzyme A, which was here. So we need this extra step to pass the shuttle to get converted to acyl carnitine. So now in the mitochondrial matrix, we have the acyl coenzyme A. And now we convert the acyl coenzyme A into acetyl coenzyme A by the third step, which contains of like different enzyme, like the four different enzymes. So now we are in this step where we have converted the fatty acids to the acyl coenzyme A, and it get transported into the mitochondrial matrix. So now from acyl coenzyme A, there is this 3-hydrogenase which required FAD and removes the H2. So from FAD you get FADH2, which can enter the electron transport chain, right, and provides you energy. And here we get a trans-enol group because there is a double bond at the trans carbon. So we get the trans-enol carbon coenzyme A, and then there is this hydratase which adds the hydrogen, and it get added to the double bond OH and H2 with the help of the enzyme trans-enol coenzyme A hydratase. And what we get is the third carbon hydroxyl group. So this is 3-hydroxyacyl coenzyme A. And then from this we use another enzyme and release two hydrogens, the 3-hydroxyl acyl coenzyme had dehydrogenase. So if you see the enzyme name, it's very simple. It's based on the substrate. From acyl coenzyme we are doing the dehydrogenation. From here we are doing the addition of water, hydratase. So from here we are doing again the release of hydrogen, which is dehydrogenase. And what we get is NADH. And this NADH can again go to the electron transport chain and can release energy. And now what we get is a keto-acyl coenzyme A. And from here we can really break this bond, right? And what you get is an acetyl coenzyme A. So if we break this bond by the thiolase, and then what we get is an acetyl coenzyme A, and this carbon molecule is released, and which can again feed into this reaction and continue the same step. So from in overall what we get is, suppose we have a palmitoyl coenzyme A, which is a 16 carbon molecule, and in each reaction one acetyl coenzyme A is released. So what we get is 7 cycle of this reaction what we need to do, right? And then from this 7 reaction what we get is 8 acetyl coenzyme A, which can enter the Krebs cycle and releases ATP and NADH. And we also get NADH, 7 NADH and 7 FADH2, which can also go into the electron transport chain and can release energy. So I hope it's clear. If not, please let me know in the comment section if you have any doubts. And now what we are going to talk about is the regulation of beta oxidation. Like what steps regulate this reaction? So the important is the fatty acids, because this whole reaction is based on the amount of fatty acids, which is controlled by glucagon to insulin ratio. So the glucagon get activated like in the when there is like less glucose available, like during the starvation condition, and the insulin get activated when there is lot of glucose present, like when we are eating and after eating, because then you have tons of glucose that you need to control. So glucagon increases the free fatty acid levels because during this, which means you need glucose, and this is a starvation condition. So during this condition, you utilize the fats that is already present. So it increases the free fatty acid levels. And insulin, on the other hand, because it get activated when you have tons of glucose, so it tries to decrease the free fatty acid level by preventing the metabolization of the already used fat. Like it says the body to store the fat, not to break down them. So it decreases the free fatty acid level. So this can control the regulation of beta oxidation. If you have more glucagon, you have more beta oxidation. And then the cat1, which is and the important shuttle, which shuttle the fatty acids, the activated fatty acids to mitochondria. It also regulates the beta oxidation. Like if you have more of this, you get more of beta oxidation. Malonyl coenzyme A, which is formed by the acetyl coenzyme A. So this acts as an inhibitor of cat1. So if you have more of malonyl coenzyme A, it inhibits the CAT1 and then you have decreased beta-oxidation. So I hope it's clear. Please let me know in the comment section if you have any doubts, and please like, share and subscribe my channel. Thank you.